Hi everybody, and welcome to our lecture on the cytoskeleton for BME2104. As the name implies, the cytoskeleton acts like a skeleton structure that provides mechanical strength for the cell, but as you'll see today, the cytoskeleton is not fixed and static like the steel girders and support beams of a skyscraper. Instead, the cytoskeleton is a highly dynamic structure that's constantly breaking down and building itself back up again. We see this especially in cell movement, which is driven by cytoskeletal growth. But even in resting, non-moving cells, the cytoskeleton will be in a constant state of flux with polymerization and depolymerization of cytoskeletal filaments. In the first half of the lecture today, we'll go over the protein building blocks of these cytoskeletal filaments and discuss the functional consequences of their polarity. In the second half of the lecture, we'll go over cytoskeleton filament polymerization, how this is controlled by a property called critical concentration, and then we'll talk about how cells can use polymerization of cytoskeletal filaments to drive cell movement. In the human disease topic that I'll use to illustrate some of these key aspects of the cytoskeleton is Listeriosis food poisoning. Listeriosis starts out as an infection of the gastrointestinal system, but sometimes spreads further through the body and can even move into the central nervous system. The infection is from Listeria monocytogenes, a soil-dwelling bacterium that becomes an invasive pathogen in mammalian hosts. In the electron microscopy image on the right, Listeria have been colored in green. You can see they have rod-shaped bacillus structure. Listeriosis is contracted by consuming raw meat or unwashed vegetables that are contaminated with the Listeria bacterium. We see around 1,600 cases every year in the United States, which corresponds to a prevalence of around 0.3 infections per 100,000 people. Most cases result in symptoms that we normally associate with food poisoning. But the disease can get much worse than this, and we see around 260 deaths per year in the United States. These deaths from Listeria infection are more common in older adults, pregnant women, newborns, and immunocompromised individuals. Being in one of these groups is a risk factor for worse outcomes from Listeria, similar to how being older, male, and immunocompromised are risk factors for worse outcomes from COVID-19. The symptoms for otherwise healthy people include diarrhea and fever, what I'd call typical food poisoning. But Listeria infection can also cause miscarriage in pregnant women due to the infection spreading beyond the gastrointestinal tract, even to the fetus. In other high-risk individuals, the infection spreading to the central nervous system can lead to confusion, loss of balance, convulsions, and meningitis. So, how does Listeria spread through the body? Many other pathogens spread throughout our body by growth and passive transport, but Listeria is different. They're able to fly around inside our cells like little speedboats, as you can see in the time-lapse video on the right. As we'll talk about at the end of lecture, the way they propel themselves around like this is by hijacking components of the host cell cytoskeleton. That's the comet tail that you see trailing behind each bacterium inside the cell. In the figure on the left, this illustrates how Listeria can move from cell to cell in our body. If they're speeding toward the plasma membrane like this, they can push out as a small protrusion. You can actually see this happen several times in the video on the right. If there's a neighboring cell immediately adjacent to this protrusion, the bacterium can get pushed all the way into the other cell, and then pinch off as a double membraned vesicle in the neighboring cell. After that, Listeria can escape from this vesicle into the cytosol of the new cell using the same proteins that it uses to escape from endocytosed vesicles in the lysosome. Using this mechanism, Listeria are able to rapidly spread through the gastrointestinal epithelium and other tissues in our body. By going directly from cell to cell like this, Listeria are able to evade many of the immune system defenses that we went over in our last set of lectures. Everything that happens on the outside of cells, like the complement system, antibodies, phagocytosis by macrophages, cell surface toll-like receptors, etc. At the end of lecture today, I'll explain which cytoskeletal proteins are hijacked by Listeria and how it's able to use them to rocket around like this. So let's talk about the cytoskeleton and how it works. I'll start out with a basic description. The cytoskeleton is a network of filamentous proteins that pervades the cytoplasm. It also includes the proteins that bind to these filaments, kind of like how chromatin includes DNA and all of its associated proteins. The protein filaments that make up the cytoskeleton are made of protein monomers that can self-assemble into the final structure, and disassemble to destroy the structure as well. What this means is that purified protein monomers can assemble into filaments all by themselves. In addition to this, they can also exhibit dynamic assembly and disassembly behavior in a test tube, which we'll talk about later. For now, though, I've organized the lecture today into two sections. In the first half, we'll go over the protein building blocks of the cytoskeleton and how they're put together. In the second half, we'll go over how their behavior is regulated in the cell and the kinetics of their assembly and disassembly. So let's start out with an overview of the different protein filaments that make up the cytoskeleton. There are three types of filaments here: microtubules, microfilaments, and intermediate filaments. All three of these are shown inside a single cell in the fluorescence microscopy image on the right. Microtubules in green are composed of tubulin proteins and play an important role as the train tracks for intracellular transport. Microfilaments in blue are formed by actin and actin-binding proteins. These play an important role in cell shape, contraction, and motility. Intermediate filaments in red are relatively stable compared to the dynamic behavior of microtubules and microfilaments that we'll discuss later. These contribute to the structural stability and strength of individual cells, as well as at the tissue level. Let's talk about microtubule filaments first. The building block for these polymers are tubulins, which make up the structure of microtubule filaments, but also have enzymatic activity. They can bind and hydrolyze guanosine triphosphate, GTP. This catalysis usually occurs when the tubulin proteins are polymerized in the microtubule structure, and the hydrolysis cleaves off the end phosphate, converting GTP to GDP, guanosine diphosphate. This switch between GTP-bound and GDP-bound states leads to a conformational change that controls the dynamic behavior of microtubule filament polymerization and depolymerization, as we'll go over in more detail in the second half of the lecture. There are 5 classes of tubulins, named alpha, beta, gamma, delta, and epsilon. We're going to focus on the alpha and beta tubulins in this class, which exist as heterodimers that form the building blocks for microtubule filaments. In microtubule assembly, the first thing that happens is that alpha-beta heterodimers polymerize into linear protofilaments, which then assemble into a sheet and roll up into a tube to form the final microtubule structure. You can see how this process works in this video from the Nogales lab. Here you can see the alpha-beta heterodimers in solution, which first assemble into protofilaments that make up this sheet and then get zipped up into the final tube-like structure, hence the name microtubules. Okay, so the sheet that gets rolled up into a microtubule is made up of 13 protofilaments, as shown in this illustration on the right. Each protofilament is made up of alpha-beta heterodimers arranged in a single head-to-tail polymer arrangement, as shown in the side view on the left with a single protofilament colored in. If you look at the same microtubule structure from the top end, this is where we can easily see that the full microtubule is made up of 13 protofilaments, which form the sheet that zips up into this 24 nanometer diameter tube. This microtubule structure is mechanically analogous to a rigid pipe. One very important aspect to microtubule structure, as you can see here, is that the entire filament has a polarity that comes from the alpha-beta heterodimers. One end is all beta, which we refer to as the plus end, and the other end is all alpha, which we refer to as the minus end. There are a couple important functional consequences for this polarity. The first is that when the microtubule filaments are polymerizing, like we just saw in that last movie, growth occurs preferentially at the plus end. This is because the minus end is stabilized and blocked, which I'll talk about more in the second half of the lecture. The other important functional consequence is that anywhere you are in the filament, whether you're at the ends or in the middle, you can always look at one of the alpha-beta heterodimers and tell what direction you're facing. This polarity will be very important when we discuss plus end directed and minus end directed motors during our next lecture on molecular motors. Alright, so that's all the basics for microtubules. Now we're going to move on to the next type of cytoskeletal filament, microfilaments, which are primarily made up of the protein monomer called actin. This is the most abundant protein inside our cells, making up over 5% of intracellular protein on average, and it plays a number of important roles in the cell, such as cell motility, which is driven by actin polymerization, as we'll talk about in the second half of the lecture today. Actin microfilaments and their associated motor proteins are also critically involved in the contraction of skeletal and cardiac muscle, which we'll cover next week. And actin microfilaments are also important for certain aspects of cell shape, like microvilli that you can see on the right. These are structures in the gastrointestinal epithelium that increase surface area to aid the absorption of nutrients, and they're stabilized by actin cytoskeleton. As I mentioned earlier, microtubules play a major role in cargo transport in the cell. This often involves vesicle transport, and I want to point out here that the actin cytoskeleton also plays an important role near the cell periphery. There's a handoff between the two systems in both directions, both for exocytosis and endocytosis. Actin filaments are different from microtubules in that they form a single double helical filament of 7 nanometers in diameter, rather than the 24 nanometer diameter tube structure of microtubules. Instead of the alpha-beta heterodimer of microtubules, actin is just a single protein monomer that stacks on top of itself to form long polymerized microfilaments. Another difference is that while alpha and beta tubulin are GTPases, actin is an ATPase, so it hydrolyzes ATP instead of GTP. We'll talk a lot more in the second half of the lecture about how the hydrolysis of these nucleotides plays an important role in the dynamic behavior of microtubule and actin filaments. Like microtubules, actin filaments are also directional. It's hard to see in this picture because the actin monomers are represented as circles, but you can imagine that each monomer is a single actin protein with an N-terminus and a C-terminus, and actin monomers are arranged with a consistent head-to-tail polarity in the filament. This is important for the myosin motors that bind to and move along actin, as we'll discuss next week in the lecture on molecular motors. The polarity, or directionality of actin filaments, is also important for their dynamic polymerization and depolymerization behavior. That we'll discuss in more detail in the second half of the lecture today. Alright, so that's all the basics for microtubules and actin filaments. The third and final class of cytoskeletal filaments that I'm going to go over now is intermediate filaments. We call these intermediate filaments because their size and diameter is in between that of microtubules and microfilaments. As I mentioned earlier, their general role in the cell is to provide strength and stability. There are 6 major classes of related intermediate filaments. I'm not going to ask you to learn all of them, but I would like you to be familiar with these 3. Keratins are found in all cells, but are especially important in the epithelial cells. As we've gone over before, epithelial cells form the outer surfaces of our body and create a barrier to the external environment. Epithelial cells exist as sheets, and keratin intermediate filaments assemble into a structure called tonofilaments that provide mechanical strength to these epithelial sheets. Next we have neurofilaments, which are important for stabilizing the axons and dendrites that branch out of neurons. These can stretch up to 1 meter in length, so it's important that neurofilaments provide mechanical strength for them. And finally, we have lamins, which form a stabilizing network of filaments on the inner side of the nuclear envelope to provide structure and mechanical stability for the nucleus. Compared to microtubules and microfilaments, intermediate filaments are the most stable and least dynamic. The polymerization of intermediate filaments is very different from microtubules and microfilaments, so let's talk about that here. Each monomer of an intermediate filament has an N-terminal head, a stalk in the middle, and a C-terminal tail. These come together to form a dimer, which still has a head-to-tail polarity. But after that, they further assemble into tetramers, and the polarity is lost here because the 2 dimers come together in head-to-tail and tail-to-head orientation. This tetramer is now symmetric instead of polar, and this is important when we consider the function of these intermediate filaments in comparison to microtubules and microfilaments. These symmetric tetramers further oligomerize to form the coiled sheet structure pictured on the right, which makes a rope-like filament bundle. As you can see from this illustration, the final rope-like filament has no directionality to it. Another way to say this is that it's not polar. Just to be clear, I'm not talking about charge polarity here. In the context of filament structures, polarity refers to whether they have a side or not. If you look at one of these filaments, can you tell the sides apart by directionality that's present in their molecular structure? Both microtubules and actin filaments have this kind of polarity, which is important because they have molecular motors associated with them that travel in specific directions, from one side of the filament to the other. But this would be impossible for intermediate filaments because they're composed of symmetric bundles that have no directionality. Okay, so we've talked about microtubules, actin microfilaments, and intermediate filaments, which make up the majority of the cytoskeleton in every cell. What I want to talk about now, very briefly, is that there are other proteins that can self-assemble and form filament structures as well. And the example I'll give here is actually myosin, the molecular motor that recognizes and binds to actin filaments. The type of myosin that forms a filament like this is a specialized isoform shown here that occurs in muscle. And it comes together with its heavy chain stalks to form this thick, bundled filament with the motor head groups sticking out. We'll go over this and the general structure of motor proteins in more detail during the lecture on molecular motors. But for now, I'll just give you our basic definition of a molecular motor. This is a protein that converts chemical energy, usually in the form of ATP, into mechanical work being force over distance, which is used in this case to move things around inside the cell, move the cell itself, or to induce cell contraction. Alright, so that covers all the different cytoskeletal filaments in the cell, and the monomers they're made up of. For the second half of the lecture, we're going to switch gears and focus on the assembly and disassembly of these filaments. As I mentioned in the beginning of the lecture, these filaments can self-assemble and produce dynamic assembly and disassembly behaviors even in their purified forms in vitro. Inside the cell, though, the assembly and disassembly of these filaments is heavily regulated. All of these cytoskeletal filaments grow and shrink by the addition or loss of their monomer proteins, and filament growth is therefore referred to as polymerization. The extent of polymerization is regulated by the concentration of monomers, the presence of accessory proteins, and by post-translational modifications like phosphorylation. Polymerization of protein filaments occurs in different stages, so let's consider each of those here. The first stage is called nucleation. This is typically slower, but once nucleation happens, that initiates the next stage of polymerization, which happens at a faster rate, as you can see here. Finally, at some point, polymerization proceeds to the point where monomer is depleted from solution. This brings us to the final steady state stage, where the monomer on rate and off rate are identical. This idea of balanced on rates and off rates for monomer polymerization is what's behind the critical concentration. The basic concept here is pretty simple. If the monomer in solution is above the critical concentration, we'll get polymerization and filament assembly. But if the monomer in solution is below the critical concentration, then we'll get depolymerization and filament disassembly. On this slide, we're going to go over a way to model this behavior mathematically, using the polymerization reaction on the right, where the reactants are a filament of length L and a monomer M, and the product is the same filament that's now one monomer longer in length, going from 7 to 8 in this example. In this system, we define the critical concentration, Kc, as the monomer concentration at steady state, where the rate of monomer addition equals the rate of monomer loss. Looking at the equations on the right, we can start out by describing the rate of polymerization, dL dt, as the sum of the on rate, K on, times the concentration of monomer, and the off rate, which is just K off. Notice an important property of the system, that the on rate changes as a function of monomer concentration, but the off rate doesn't. That's because the off rate is just based on polymer ends, and the number of polymer ends stays fixed here, it doesn't change. If we set dL dt to 0, we can derive the concentration of monomer at steady state pretty easily. This is just K off divided by K on, and this is how we define the critical concentration, Kc. As a quick note, the units of Kc are in concentration, because the units of K off are moles per second, and the units of K on are moles per second per molar concentration. This makes sense if we consider the on rate for a second, because as the concentration of monomer M increases, we expect the on rate to increase as well. What this means for the critical concentration is that if the concentration of free monomer is greater than Kc, we'll get polymerization and filament growth. But if the concentration of the free monomer is less than Kc, we'll get depolymerization and the filament will shrink. Everything we just talked about for the critical concentration may seem straightforward and simple, but where things get interesting is when the critical concentration can change dynamically, as it does with tubulin, depending on whether it's in the GTP or GDP bound states. And this leads to some really interesting and complex dynamic behaviors for microtubule filaments. Let's go over how this works here. Starting out, remember that alpha tubulin and beta tubulin heterodimerize. That's what we see in the top right corner of this illustration on the right, those little green figure 8s. For the purposes of our discussion on this slide, I'm going to refer to this alpha-beta dimer as a monomer, because that's how it behaves with respect to polymerization in microtubules. So the alpha-beta monomer binds GTP. And remember, in addition to being a structural protein, tubulin is also an enzyme with GTPase activity. But as a free alpha-beta monomer in solution, the GTPase activity of the individual tubulins is relatively low. It's only once it becomes polymerized into the microfilament that the GTPase activity of tubulin is really turned on. This is caused by a conformational change induced by protein-protein interactions in the filament, and now GTP is rapidly hydrolyzed. You can see that in the picture on the right. Green alpha-beta monomers are GTP bound, and the yellow alpha-beta monomers in the filament are GDP bound, having hydrolyzed their GTP after binding into the filament. As more GTP-bound alpha-beta monomers polymerize at the end of the microtubule, we get polymer growth, and the end of the microtubule remains green and GTP-bound. This is called the GTP cap. I put it in green here to match the diagram, but you should consider this a blue term to focus on while you're studying. As long as filament polymerization happens faster than the rate of GTP hydrolysis, the end of the filament cap will remain GTP-bound with this GTP cap. But if the rate of polymerization ever slows, perhaps because the concentration of local alpha-beta monomers is depleted, then GTP hydrolysis will catch up and reach the end of the filament, and we won't have a GTP cap anymore. You can see that happening in the figure on the right. The thing that's really interesting about this is that the GTP cap has a lower critical concentration than the rest of the microtubule, which is GDP bound. And what this means is that when the GTP cap is lost, the microtubule rapidly disassembles in a process called catastrophe, because we're suddenly in a regime where the critical concentration needed to maintain equilibrium is much higher, and the actual concentration of free alpha-beta monomers in solution is way lower than this, so we get rapid depolymerization and disassembly. As a consequence of these properties, microtubules exhibit a behavior called dynamic instability, which basically means that they're constantly going through cycles of growth and then rapid disassembly. Even if we just take purified alpha and beta tubulin and put them in a test tube, they'll exhibit this growth and catastrophe behavior, as long as we add in enough GTP. This dynamic instability behavior turns out to be really useful for cells. One nice advantage to this behavior is that once catastrophe happens, this frees up a ton of alpha-beta monomers that can be used to seed or polymerize many new filaments. Just to make a quick point about how this works, once the GDP-bound alpha-beta monomers dissociate from the polymer, this causes a conformational change that favors GDP dissociation and GTP binding. This is how all these alpha-beta monomers are turning green again. Here I'm using the same color scheme as on the previous slide, where green equals GTP bound. In this time-lapse video of a single cell in culture with fluorescently labeled tubulin protein, we can see this dynamic instability behavior in action. It's just a short clip, but for several of the filaments, you can watch their growth followed by rapid disassembly. After catastrophe, new microtubules grow back in a different, seemingly random orientation, which allows them to explore and cover the entire volume of the cell over time. This coverage is useful for the cell because these microtubules serve as the tracks for vesicle transport. The fact that these tracks are always shifting and changing to cover the entire cell volume means that every single location in the cell is able to have vesicles delivered to it. So where do these tracks come from, and where do they go to? The most common arrangement is to have a microtubule minus ends in the middle of the cell, and the plus ends at the periphery. The minus ends are typically fixed in place and stabilized so they don't depolymerize, and the plus ends go through the dynamic instability stages of growth and catastrophe like we see on the left. Every cell has a structure close to the nucleus called the centrosome, which acts as a microtubule organizing center, and orients the microtubules with radial polarity like this, and their plus ends facing out towards the periphery. Microtubule organizing centers, or MTOCs, nucleate microtubule polymerization within the cell, and form stable minus ends that don't experience further polymerization or depolymerization. As a consequence of this, only the plus ends of the microtubule are exposed. As we see on the previous slide, the centrosome is an MTOC that's present in all cells close to the nucleus in the center. The centrosome is made up of two tubulin structures called centrioles. You can see the same structure here by electron microscopy. This MTOC structure will serve as an anchor for microtubules in different contexts that I'll go over on the next slide. As you'll see, there can be other MTOCs in the cell, in addition to the centrosome. In neurons, microtubules play a very important role in transporting cargo such as neurotransmitters to and from the ends of axons and dendrites, these long extensions from the cell body that are critical for neuronal communication, and are actually heavily branched at the ends, unlike what we see in this simplified diagram. The axon of some neurons can grow up to a meter in length, so it's very important to have active transport along this structure, because relying on diffusion alone would never work to get cargo from one end to the other. This transport of cargo over long distances in neurons will actually come up next time in our lecture on molecular motors. We'll see how this transport system is hijacked by different viruses, and then I'll explain how neuroscience researchers have hijacked these viruses to use them as tools for tracing neural circuit connectivity. Another important cell type that has specialized microtubule function are these ciliated epithelial cells. Cilia are microtubule-containing extensions that increase the cell's surface area, but also sway back and forth in a wave-like manner to help move particles and fluid along the surface. The next example of specialized microtubule function is in red blood cells, where they form marginal bundles that wrap around the perimeter of the cell and contribute to its unique donut shape. In every cell, the microtubule network acts as a distribution system for cargo transport, with some motor proteins that carry their cargo towards the periphery and others that are directed towards the center, specifically the centrosome microtubule organizing center. This microtubule network rearranges during cell division to form the mitotic spindle during metaphase, and this structure serves to arrange all the chromosomes along the metaphase plate and then separate them during anaphase. We can see this process happening here in another cell culture time-lapse video with fluorescently labeled microtubules, this time looking at a cell as it undergoes mitosis. We'll talk about this process a lot more later in the semester during the lecture on cell cycle. But for now, let's just focus on the role that microtubules play in chromosome segregation. When a cell is ready to divide, this means that it's replicated its DNA and also its centrosome, and now the two centrosomes migrate to opposite sides of the cell. The replicated chromosomes condense to their most compact state in matching pairs, and then are pulled to the middle of the cell by the two microtubule networks that are seated by each centrosome. At this stage, the microtubule network is referred to as the mitotic spindle, and the centrosomes are referred to as spindle poles. The microtubules attach to each chromosome pair with their plus ends at a structure called the kinetochore, which is located at the centromere region of each chromosome. Once the cell is ready to divide, the proteins holding the kinetochore together are cleaved by a protease, and then the chromosomes are pulled to separate sides of the cell by the mitotic spindle and specialized motor proteins at the spindle poles, which we'll talk about in more detail in our lecture on the cell cycle. Because microtubules play such an important role in mitosis during cell division, any drug that disrupts microtubule function will interfere with this process, and this can be exploited for anti-cancer therapy because cancer cells grow and divide much faster than most normal cells in the body, and will therefore be harder hit by these inhibitors, which are called antimitotics because they block the progression of mitosis. Generally speaking, drugs that target rapidly dividing cells are referred to as chemotherapeutics. This is what people are referring to when they talk about getting chemo for cancer, and all the side effects like hair loss and nausea come from their effect on non-cancerous cell types in our body that are also dividing at a faster rate, like in hair follicles, for example. We have many different examples of antimitotics that interfere with microtubule function in different ways, and all of these can be used as chemotherapeutic drugs. The drug vinblastine causes tubulin aggregation, clumping them all together in aggregates so that alpha-beta monomers can't be used to polymerize new microtubules. Nocatazole promotes microtubule depolymerization, preventing the mitotic spindle from forming. And Taxol actually stabilizes microtubules, blocking cell division by freezing and locking the mitotic spindle in place. Many of these products were originally identified in plants, such as the periwinkle flower and the yew tree. Natural products like this are generally larger and more structurally complicated than compounds we can synthesize in lab. For the most part, Mother Nature is still a better chemist than we are. Okay, in addition to pulling chromosomes around in mitosis, like we were just talking about, microtubules are also used to produce cell movement. On top, we have a description for the power stroke and recovery stroke of beating cilia that we talked about previously. This is used to move fluid and particles along the surface of epithelial tissues and is powered by a molecular motor positioned at the base of the cilia microtubules. On the bottom, we have a cell that's actually motile itself, flagellated sperm. Microtubules make up the structure of the flagella, and a molecular motor positioned at the base of the flagella is connected to the microtubule structures to produce the whip-like flagellar motion that propels the sperm cell forward. Alright, that's all for microtubules. For the rest of the lecture, we're going to talk about the regulation of actin polymerization dynamics. Just to remind you, I'm showing the structure of an actin filament again, with its plus end on the left and its minus end on the right. These are both determined by the orientation of actin monomers in the filament. Unlike microtubules, which are always stabilized at the minus end by the microtubule organizing center, actin microfilaments can be stabilized at either end, or both ends can be free. A couple more things to go over here. Unlike tubulin, which is a GTPase, actin is an ATPase. The behavior is very similar though. Free actin monomers tend to have ATP bound, and when they polymerize, their enzymatic ATPase activity is turned on, causing ATP to be rapidly hydrolyzed into ADP. Another thing is that for naming conventions, the plus end is referred to as barbed and the minus end is referred to as pointed. I'm mentioning this now, but it'll make a lot more sense when we see this again in the molecular motors lecture. One last thing to explain on this slide is G-actin and F-actin. We won't use these terms in this class, but just to make sure you're not puzzled by what they mean in this figure, F-actin is for filamentous and G-actin is for globular. That's the term we use for round, blobby-shaped proteins, which is pretty typical for soluble cytosolic proteins. Okay, let's talk now about the properties of actin polymerization. The plus end of an actin filament grows faster than the minus end because the critical concentration, Kc, is lower at the plus end. This difference in critical concentrations comes from the polarity of the actin filament. If you were to look at each end, they'd look different. One end has one side of the actin monomer, and the other end has the other side facing out. On top of this difference in critical concentrations, actin's ATPase activity is activated in the polymerized filament. Similar to microtubules, we have ATP at the end where the filament is growing fastest, and ADP everywhere else. The difference in critical concentrations is accentuated by the ATP versus ADP states of the plus and minus ends. These properties of actin polymerization lead to an interesting behavior called treadmilling. The basic idea here is that polymerization at the plus end and depolymerization at the minus end will make it look like the actin filament is moving in the plus end direction. And it is in a way, but if you were to track and follow any one of the monomers in the filament, you'd see that each one is just standing still. The arrows in this figure illustrate another interesting aspect to this behavior. The free monomers diffuse randomly, but the net movement of monomers by diffusion is toward the plus end, because we have a source for monomers at the minus end and a sink for monomers at the plus end. In this way, many of the monomers will move forward along with the filament as it treadmills forward, completing the treadmill loop. Alright, so that's how actin monomers behave on their own in a test tube. But inside a cell, things are much more controlled and regulated. There's a whole set of proteins that control every step of this process, and I'll go over the most important players here on this slide. Using the same color scheme as on the previous slide, we'll start out with free actin monomers, which are blue for GDP bound. The first thing that happens is we have profilin. This is a shuttle protein that also promotes nucleotide exchange. If you remember the GEF and GAP proteins that we talked about for nuclear import and export with RAN-GTPase, this is the same idea here, acting as an exchange factor that causes ADP to dissociate, and that facilitates a new ATP molecule coming in to bind. Next, we have thymosin, which acts as a monomer buffer. What this means is that it binds actin ATP monomers and sequesters them away, keeping them from polymerizing into actin microfilaments. This may seem a little counterintuitive, but the idea here is that this provides a way for the cell to store up actin monomers, which then can be used all at once when they're needed for rapid polymerization. This is particularly important because actin polymerization is used to drive cell movement, and these rapid bursts of monomer are what allow the quick movement and changes of direction that we saw in the video of the neutrophil chasing the bacterium in our lecture on PAMPs and pattern recognition receptors. On top of acting as a nucleotide exchange factor, profilin also acts as a shuttle that helps facilitate the addition of actin monomers to the microfilament plus end, like this. Profilin is regulated by many signaling pathways in the cell to help control when actin polymerization is turned on or off, and part of this is through competing with thymosin for actin monomers. In addition to profilin and thymosin, we also have cofilin, which acts as a buffer for ADP actin, gelsolin, which acts as an actin severing protein. This generates more plus ends and therefore can actually increase microfilament polymerization. And then we have the cross-linking proteins alpha-actinin and filamin, which help provide more structural stability to the actin mesh network. So as I mentioned earlier, most types of cell movement are driven by actin polymerization, just like we see in this video of a neutrophil chasing a bacterium. As the actin cytoskeleton is pushed forward and out, this pushes the cell forward. On top of that, we need to have depolymerization of the actin cytoskeleton on the reverse side of the cell, so it can squeeze in and keep the whole cell moving in the forward direction. This entire process is coordinated by local signals coming from the bacterial protein sensing receptor, FBR, and also how far these signals can diffuse through the cell from one side to the other. Generally speaking, the structures made by actin cytoskeleton pushing out during coordinated cell movement are referred to as lamellipodia for a sheet-like foot, and pseudopodia for fake foot. That's really more like a little arm reaching out. In addition to neutrophils, another cell type that we see this behavior in is fibroblasts. Remember, these are the cells in the interstitial space that migrate to the site of wounds and produce large amounts of ECM material to help with wound healing. We also see cell motility and this crawling-like behavior in cancer. Gaining this ability for cell movement is often the step whereby cancer cells become malignant, meaning they can migrate from the primary tumor to other parts of the body. In addition to all the natural product drugs that block microtubule function for cancer therapy, we also have natural products that block actin microfilament function. But these aren't used as anti-cancer drugs. They're just generally toxic instead, killing all the cells in our body, whether they're fast dividing or slow dividing. Phallotoxin is an actin filament stabilizer that's the poison in some types of mushrooms like death caps pictured here. This will mess up pretty much all the cells in our body, but the most acute immediate cause of death in phallotoxin poisoning is liver failure. An interesting story about this molecule is that it's also used as a reagent for fluorescence microscopy. If we conjugate a fluorophore to phallotoxin, or phalloidin as we refer to it when it's used as a research tool, this can give us beautiful cell images, like we saw in slide 6, for example. Another natural product compound that interferes with actin microfilaments is cytoskeleton. Like noconazole for microtubules, cytoskeleton is an actin filament destabilizer, also derived from mushrooms. All of these microtubule and actin-inhibiting natural products have generally arisen evolutionarily as defense mechanisms against pathogens for plants and fungus. Alright, let's finish up with a few more details about actin microfilament polymerization at the leading edge, which is how we refer to the plasma membrane that's moving forward during cell movement. I'm going to introduce two more regulatory proteins here, ARP23 and WASP. ARP23 is a nucleating protein, which binds to existing actin microfilaments and seeds new filaments at an angle that's approximately 70 degrees from the original plus end. We can think of ARP23 kind of like a cross-linking protein, except instead of linking two fully formed filaments together, it just nucleates the new microfilament instead. And the additional actin polymerization in nucleates at 70-degree angles like this helps drive net flux of actin filaments towards the plasma membrane. What WASP does is relay signals from cell surface receptors, like the FPR for example, and convert this message into activated ARP23. To give you an idea of what this cross-linked actin network actually looks like near the periphery of the cell at the leading edge, here are two electron microscopy images at different levels of resolution. As you can see, the actin filament network here is extremely dense. Generally speaking, cells will have a thick shell like this all the way around their surface in a sphere, which contributes to the mechanical and structural integrity of the cell along with the intermediate filaments. And now that we understand all of these components, we can come back to Listeria monocytogenes and explain all the details about how this bacterium hijacks the host cell cytoskeleton to propel itself around the cell. Its trick is that it has a protein called Act-A on its surface, but only on one side of the bacillus rod shape. This polarity of Act-A on the Listeria surface is critical for its movement, as we'll see in a second. What's special about Act-A is that it has two different domains that nucleate actin polymerization, an ARP23 recruiting site and a WASP-like domain that activates ARP23. By dramatically increasing the local concentration of activated ARP23 on one side of the bacterium, this seeds new actin microfilament polymerization, and then additional branched polymerization on top of this, pushing the bacteria forward through the cell, just like the plasma membrane is pushed forward during cell movement. We can also take polystyrene beads and coat them with Act-A protein to see a similar effect in vitro. As long as we provide all the components like ARP23, actin, ATP, etc., we can get polystyrene beads rocketing around on a microscope slide. The one trick here, though, is that we need to make sure we only coat one side of each bead with Act-A protein. If the beads are coated with Act-A all the way around on all sides, then we'll get actin microfilament polymerization on all sides at once, and each bead will just sort of fuzz out in all directions, without actually moving in any one specific direction. This experiment highlights the importance for Listeria bacterium to express Act-A on just one side of its rod shape, rather than all over its surface. Okay, so now we know how Listeria moves around in our cells by hijacking our own cytoskeleton proteins. Is there any way we can use this knowledge to treat listeriosis? Unfortunately, so far the answer is no to this question. The best treatment we have is just time and lots of fluids for healthy individuals, with the addition of standard broad-spectrum antibiotics for immunocompromised individuals and pregnant women. The problem with any specific mechanism-based treatments that target ACT-A, ARP2/3, or actin polymerization is that we'll likely see the same effects that we saw for the mushroom toxins phytotoxin and cytoskeleton. Because actin function is so important to all the cells in our body, any treatment that disrupts this will have very bad toxic side effects. It seems plausible that some kind of inhibitor could specifically target ACT-A and not wasp protein in the host cell, but this doesn't seem to be the case yet. We haven't discovered this, so for now we just stick with a simple treatment of time and rest. To reduce the risk of Listeria infection in the first place, we can avoid eating specific foods like soft cheeses, refrigerated meat and fish, and we can always practice good food safety and hygiene. A key thing to remember here is that Listeria is everywhere, and we're always eating small amounts of it with our food. As long as we wash off the food well and avoid riskier foods like in these categories, then the amount we ingest is small enough that our immune system can fight it off. As I mentioned earlier, the fact that Listeria can move from cell to cell in a membrane-enclosed protrusion means that it's shielded from the extracellular components of our immune system, like macrophages and antibodies. But it's not completely invisible to the immune system. It can be detected by cell internal PAMPs and the T-cell response to infected cells, and this is how we eventually fight it off. If you're interested in the cytoskeleton and cell migration, then I recommend checking out the work of these UVA faculty members. Jim Casanova, who studies the role of ARF-GTPase in cytoskeleton assembly, Doug DeSimone, who studies the relationship between integrin signaling and cell migration, Dorothy Schaefer, who studies the protein dynamin-2 in actin network migration, and Tom Barker in our own department, who studies cell migration and fibrosis. Alright, that's all for today. In our next lecture after the exam, we'll move on to molecular motors, and you'll see a lot more details about how the cytoskeletal filaments we covered today serve as tracks for cargo transport. Thanks for watching, everyone. Good luck studying for the exam, and I'll see you next week.